The camera will pick it up from here. So.
I hope it's not that high quality of a camera.
It's pretty far away. So, good afternoon. Welcome to the Faculty Lunch and Learn session. Today's topic is going to be on getting started or setting up a lab here at U of A, and then how to recruit and mentor students. Our first presenter is Dr. Mark Beilstein, and he will take the floor in just a moment. He'll be followed up with Dr. Rachel Gallery about halfway through the presentation. With that, I'll turn it over to Mark.
Great, thanks, Chris. It's actually Beilstein, just for the record.
Yeah, it's okay. My bad.
Like the famous chemists. Yeah, do you know Beilstein Institute? Flame test? None of this? Wow, you guys aren't very good journalists. So I am a faculty member in the School of Plant Sciences. And of course, Rachel is also a CALS person who's coming up later, Rachel Gallery. So she's in the School of Natural Resources and the Environment. And I think we do a pretty good job in CALS of getting undergraduates to work in our group. And I've been at the U of A now since 2011. And I have had about 30 undergraduates come through my lab in that time. And the nature of our research, of course, requires just some undergraduate hands to help out. But in addition to that, a major goal for me is to train the next generation of scientists in scientific inquiry. So obviously the place any of us begins thinking about adding undergraduates to our group is basically assessing our needs. Like what do we need to get done in the lab that would be under the purview of an undergraduate? And that might be different for a lot of different people. I know, for instance, that we have a series of menial tasks, what I would call menial tasks, that are absolutely essential to be done in the lab. And that includes for us primarily plant care. So we need someone to simply grow the organisms that we work on. And this requires a lot of attention to detail. It requires someone to be very schedule-oriented. You know, they need to have excellent follow-through. And that's kind of the baseline, right? You don't have to be a rocket scientist to grow plants. A good green thumb helps. A good attention to detail helps to understand what's going on, to observe the plants when you're growing them, to understand what's going on. But it's the type of task that just about anyone could do. And so, you know, the answer to the question of involving undergraduates at that level might not be, oh, I need someone who's really interested in plant biology to complete this task. It might be simply, hey, I need to pay someone who has some work study hours that needs to get involved somewhere across campus to help them pay their way. And that might be the right opportunity for somebody, and it might be the right match for the lab as well. And so that's a consideration at first. It's like, are you requiring someone who is really interested in the research you're doing, or do you require just hands to get a task done? Things like the equivalent, our equivalent of washing dishes, essentially, which is watering plants. I actually, incidentally, in my own lab, I don't have positions like that, but I know plenty of my colleagues who do. So even the people who water the plants for me are people who have expressed an interest in the lab, and they are starting with that absolutely critical task. But it's quite possible you don't need a student researcher to say, do some general task in your lab. And then, of course, if the answer is, well, I do want someone who is interested in research, then you have to ask yourself, what are really the qualifications that are going to lead to a successful laboratory experience, both from the aspect of the student as well as the aspect of the lab. So for me and all of the individuals that work with me. And in addition to that, I like to ask myself, well, what are the broader goals for including some undergraduate in my group? For me, these are just a couple of off the top of my head ideas that I have which are part of my assessment of the needs of my lab, are the ability to provide mentoring opportunities for the folks in my lab who are either my research staff or the postdocs or graduate students in my group. And I would even put talented undergraduates in that as well, because I'm trying to train them too. And really important aspect of that training is to provide an ability for them to manage their own projects, which means managing people. I think you hear it said a lot by faculty members. You know, you're never trained for this job before you step into it as a faculty member. And at the same time we're saying that, it's important that we do something to remedy that problem. And I think part of that is teaching people how to manage not just themselves, but how to manage other people in the lab. And so to provide those opportunities for them not only to mentor them, but also to schedule out what it looks like to utilize the hands that you have available to you over a reasonable period of time to get a job done. Like, really important aspect of becoming a scientist who's going to lead projects instead of a scientist who's just always going to respond to the needs of a particular project. And of course, a huge part of it for me is to inspire those students with that hands-on experience that they absolutely can't get elsewhere aside from being completely integrated into a laboratory group. And part of that hands-on experience and excitement and exposure to scientific inquiry is exposure to the folks who are also working in the lab. So what does it mean to have a career in research? It's more than simply doing the experiments. It's interacting with the group, sharing your ideas, etc. And that part of the experience is incredibly valuable as well. So I asked myself these things and said, okay, what is it that I'm trying to accomplish by adding another undergraduate? And I want to make sure that I can hit on these goals and do it thoughtfully in a way that's actually not going to detract from the dynamics of the lab or my ability to mentor the people who are working for me, who are, again, research staff or postdocs or graduate students. So I don't want to overwhelm them, is what I'm trying to say, with too much mentoring or with too much project management. I want to make sure I hit that fine spot. Of course, a major issue that everyone faces is, where do I find these undergraduates? This holy grail of the talented, bright undergraduate who is going to actually contribute something to the lab rather than suck up everyone's time and energy and resources, aside from time and energy, just in the molecular biology lab, the resources that are required to do it. So, of course, you know today that there is an Office of Undergraduate Research. That's why we're all here. And if you speak with Chris about the opportunities that are available through through the Office of Undergraduate Research, you'll learn that there are resources for matching students with researchers, and that those resources are growing. So I think it's always a good idea to check back in with Chris and get listed on the website that indicates that you may have availability in your lab whether you're looking for students. Another great resource I don't want you to overlook is your departmental advisors, so your undergraduate advisor, as well as the undergraduate advisors of related departments. So if you can establish a relationship with these people, reach out to them and talk with them and let them know about opportunities. They often are having conversations with students who are really excited about doing some kind of research but have no idea where to do that research. And they are more than happy to try to match students with interested labs. So I know the departmental advisors in the School of Plant Sciences. We have a couple of them. And again, they can not only give you an idea of which students are looking, but also help you figure out something about those students, right? So that you're not going in blind. They often have a good understanding of their undergraduate record, as well as what their interests are and whether they have any related experience. So it's a fast track to information. Of course, the undergraduate biology research program on campus has got a pretty big footprint here, and you can get listed on the UBERT website as well, and you just become affiliated with the program and mention that you're looking for students. And this is also a clearinghouse information for students, so they can go and look at different labs and the individuals who are looking for undergraduate researchers within this program. And I won't take a lot of time to explain what the UBERT program is, because there's a lot of good resources on campus for this, but I will show you how I integrate UBERT later into the way that I filter students coming into the lab. Again, this program has been absolutely essential for my research. I have had more than 50% of the students who come through my lab end up as UBERT fellows. And it not only helps pay the bills, which is great from my perspective, but it also gives them additional training and some ability to develop soft skills outside of the laboratory. So I'm not the only one, or my lab isn't the only group responsible for mentoring them in some of these other important aspects of being a research scientist. And then, of course, I want to encourage everyone to get involved in introductory courses within your discipline or related disciplines. This is how I find the majority of my students, and I mean, I'm cheating because I teach one of these introductory courses, but it's a great idea to identify some of the introductory courses that would attract students interested in research that you're doing, and speak to those instructors and ask, hey, is there a time, can you show me your syllabus? Turns out I'm an expert in X or Y. I would be really happy to come in for 5 or 10 minutes and just tell them something about this kind of research, what's happening at the U of A, integrate it with maybe recent findings or things that are happening locally, nationally, globally, that will pique their interest. And then it's a short plug for you to say, hey, if you're interested in these questions, I'm also running a lab where we ask about these questions and try to find solutions to the problems that are outlined. So again, it's a great way to identify students early in their academic careers because you're getting them in an introductory course. And if you give this short presentation, you'll typically find that students are starving for some kind of hands-on experience, and you'll get a lot of responses. And then it's a job to filter those responses, but in association with the instructor, you can ask then, well, can you tell me, could you identify, if you were choosing students for your own lab, could you identify some of these students who you think would do well in a laboratory environment? And I can show you how I use that information. Of course, I have access to all of their grade information, which is something that you may not want all that grade information, you probably don't want all that grade information, but an assessment by the instructor is just as valuable. One of the things to remember to do during this 5-minute presentation when you talk with students is to really provide a clear outline of your expectations in terms of what you're looking for. This will help filter out students who, it sounds great, but then, oh, if I can't do X or Y, then it's not the appropriate experience for me. So think about what you really want. And if it's like the case where the student says, oh, well, I can only work 5 hours a week. Okay, well, that typically doesn't work in my lab for a laboratory experiment. If you can't put in roughly 10 hours a week, it's not worth it to us to try to train you because, and it's also not worth it from the student's perspective because they won't actually accomplish anything. They'll never be able to put all the pieces together because they're just not there frequently enough. So if that's something that's a deal breaker for you, you have to outline it in a short presentation. So think of 3 or 4 key criteria and present those in that 5-minute vignette about your research availability or the available research in your group. Okay, so this is how I do it. I teach Introductory Biology 1, that's MCB 181. It covers cell and molecular biology. That's great for me because I'm a cell and molecular biologist. It has about 350 students each fall semester. That's just in my section, but there are about 1,800 students overall across 5 different sections. In week 5 of the course, we cover photosynthesis, which is great as a plant biologist. And at that point, I like to hit home to students how absolutely essential photosynthesis is to the sustainability of our lives. And to mention that, this is one of the reasons that I love being a plant biologist, is that I'm working on these incredibly essential problems that affect whether or not we can survive on the planet. And then at week 12, we hit DNA replication, and this happens to be an opportunity where we can talk about telomeres and telomerase, which are 2 other topics of inquiry in my group. And so at that point, I'm actually lucky here because telomeres and telomerase, this particular topic area is one that fascinates students. And so you can take their natural interest in this kind of bizarre end replication problem and say, hey, you thought that was really cool and interesting, and it has to do with cellular aging, and it has to do with senescence of cells, and also has a relationship to cancer. It just turns out we also study those same processes in plants. And so if you have a real interest there, then you should let me know, and I'm going to hire a couple of students out of this class specifically to work in my lab. And at this time, I set out these criteria. This is what it's going to take to be required. This is what it's going to require to be considered for any of these positions. Okay, so what are the requirements? Try to keep it pretty basic. You have to get an A or a very high B in MCV 181. You have to be willing to work for credit for 1 semester. And I typically don't take volunteers. I require that students work for credit during that first semester. After the first semester, I tell students, if you like it and we like you, then there's an opportunity to continue over the summer. You'd be invited to join the lab as a full-time undergraduate researcher over the summer, and that would be a paid position. Yeah, correct.
Can you say a little bit about why you don't want students volunteering, or why you don't accept volunteers?
Because one, it's going to require a bunch of work for them to do it, to have a real research experience. 2, there are a number of additional deliverables that I'm going to make everyone do, whether they volunteer or they don't. And so they're getting the same, if they come and volunteer, it has to be the same experience as someone who's getting credit. And so they should just take it for credit, because those credits will help. So, and it locks them into a certain number of hours per week. It's like that, these are my expectations that you will perform this way. Even if you think you're volunteering, you're gonna have to live up to these expectations. So you might as well just take it for credit. So I want it to be a serious experience for them. And I like the fact that there's an obligation to show up. If I'm paying them, it's the same thing, right? It's just that in this case, they're paying me, kind of. It's the first semester, so it's going to be a net input on our part during that first semester. Other questions about those criteria? So those have been working for me to identify students over the last several years, but you have to just think about what the criterion might be. I used to have a former postdoctoral advisor who told me your very best students are the high B students, the ones who just missed the A, because they still have something to prove to you, which is an interesting sentiment, but I think the best undergraduate I ever trained in her lab fell into that category. So I think for some students, that's absolutely true. Okay, so what do? At the end of the semester, I remind students of the opportunity and ask they email me if they're interested. I collect all those email inquiries. I schedule an informational meeting. I do this during the first or second day of the spring semester. And then I invite all those interested students who meet the criteria outlined to attend the informational meeting. And I also ask my colleagues in the College of Agriculture and Life Sciences to attend those meetings and consider presenting their research. So what do we do at the informational meeting? I invite researchers from across CALS who are looking for undergraduate researchers with an interest in the biological sciences. They come, they attend the meeting. I ask they give a short 10-minute presentation about their research, and the meeting works something like this. Students introduce themselves and they provide a brief bio, so they tell you what year they are, what their major is, maybe what their career goals are. We go around the room. Typically, I have between 10 and 20 students. So it's culled pretty well by, out of a class of 350, it's culled pretty well by the grade requirement and also the requirements to work for credit over the semester, and a large one is to work over the summer. So I tell them, I think I maybe missed that a little bit on the previous slide, but I tell them you need to be able to work over the summer. If we like you, you like us, you will get a paid position over the summer. But if I'm going to invest in your development over the spring semester, then you're going to have to put in time over the summer.
Mark, but that's not for the students that are getting enticed and working in your lab in session number 5 or 12, week number 5.
Oh, I just that was just a schedule of when I introduced the idea in the semester.
I'm confused because many other researchers in CALS are not working on television.
Right.
So.
Oh no, so that's just me. This is how I do it for me.
Wait, but you're inviting researchers and us.
I'm enticing students, particularly from my lab,
Right.
but there's 20 of them, and there's no way I can take 20.
Good enough.
So that's a great question. So my goal is get them in the door by getting them interested, telling them the types of things I do. And then at the same time, because I can't accommodate everyone, I used to do it just me, right? And I would interview maybe 10 or 12 students, and I would turn away the majority of students and keep a couple. And then I realized, well, I'm turning away really great students, and I started asking the faculty on my same floor in Marley. And then over the last couple of years, I've expanded beyond that, and I've asked Sangeeta Pawar, who's the Associate Dean for Research, or the Assistant Dean for Research in CALS, if she would help me identify other researchers in CALS who would be interested. And I sent out a blanket email to those folks inviting them to come, and they get back in touch with me, and then we go through this protocol. Yeah.
So it could be conceived of that those 30 students that really don't get enticed by the telomere stuff, where you get 20 students, those 30 students would actually benefit if they learn also a little bit about what the faculty in CALS do to come to the informational meeting.
Absolutely.
Where they wouldn't be trapped in telomerase.
Absolutely. Yeah, no, absolutely. No, and of course, my goal started out being get some really great undergraduates for my lab, and it's expanded to, okay, let's see how many research opportunities we can provide as a faculty across the college for these very talented students. So, and I think many other avenues of using, a similar strategy, but casting a broader net would work.
Just because so many students, especially in the introductory course, have no idea what's going on in agriculture. Right? That's
Yeah. That's so incredibly true. They have no idea what's going on. Okay, so students introduce themselves, they give a bio, faculty give a short interactive presentation on their research. I ask that faculty focus on impact, because most students want to know that the things that they're doing are answering big questions, and you won't get them by droning on about the details of some process that they're only vaguely familiar with. You'll get them with impact. Let's work on this problem so that we can make solutions that will make the world a better place. Is a better strategy. I give students a little score sheet with the names of all the presenting faculty, and they're asked to rank them then after the presentations in order of their interest. And then I provide the faculty with the names and email addresses of the highest matched students for their research. And then the rest is up to folks who are in attendance to follow up, send emails, and set up additional interviews at some point if you want.
When do you normally do that?
So that the meeting will be that first couple of days of spring semester, because we are getting into kind of a timing issue here, and I'll show you what I do. And you can see why I'm in this bit of a timing issue. So each fall semester, I ask myself, well, what are the needs in the lab based on the current composition of the lab, especially people who are graduating either in the winter or in the spring. And I ask myself, of course, two important questions. What projects in the lab will require additional hands once those other students graduate? And who's available to mentor additional students? And this becomes that really important question for me to hit the right sweet spot between the total number of students that we can effectively mentor with the available mentors that I have. Right? I don't want to overload people with too many students because then it's not a good experience for the student and it's not a good experience for the mentor. Right? So I have to figure out what the mentoring capacity of the lab is, and I will not accept, so you end up with a lot of students who say, oh, I want to come do this. I could just volunteer, or I could do, I could come by and do X, Y, or Z, and I invariably say absolutely no to that. I don't let anyone walk in the door that I can't put with a person who will teach them how to be a good plant molecular biologist, because otherwise, it's just diluting out, one, the experience, and two, the time and energy of the people who are involved in mentoring, right? It's a really big responsibility for the person who's directly mentoring that student, and it won't be me. So, there's just not enough time in the day for me to do that. So typically, over the last several years, I've required 2 or 3 students each January. And of course, once you kind of get in once your lab is of a particular size that meets the project needs, you kind of get into this flow where if you have a couple of students who join every January, then a couple years later they graduate, and you need 2 more to replace them. And so that's kind of the flow that we're in right now. So I'm, of course, one of the researchers that they can rank, and then I will follow up with all those who rank me as their first choice and schedule a second interview. And the second interview, they come in and we sit down and just chat a little bit. I usually know them pretty well by that time because they've taken the course from me. And typically it's the better students, and we've had multiple conversations either about research or about content in the course. So I don't spend a bunch of time asking them a gazillion questions. At this point, I feel like, okay, they're qualified to be here. The next question is, do they fit in the group? So that is an incredibly important question for me to know the answer to as best as it can be assessed. And so I sit them down with all of the other folks in the lab. So I have a brief chat and I say, hey, I want to take you to the lab. It's where you can meet everybody, figure out whether or not this is a good match, whether you feel like this is going to be a group that works well together. So after the lab has met everyone, typically I'll interview 4 or 5 students for the 2 positions. After the lab's met everybody, then we all sit down at a post-interview meeting, and they tell me what they thought. They say who they like best. I allow them to talk to me individually to solicit feedback in case they don't feel like there's something that maybe doesn't sit right, but they don't want to share in front of the entire group. And I share my own perspectives about the students, and then together we come to a collective decision about who is going to enter the lab. So we do it as a group because it's a group that has to work together. And I'd have to say that strategy up to this point has worked really well, and that they're a pretty cohesive group that enjoy being around each other and can work together. And I think that's partly because they've had that ability to assess from the very beginning, and that input there is the people in the lab are just as invested as I am in the success of this individual because it's their choice too. I think very rarely have I chosen differently than they. It has happened. It has happened. Very rarely. And only if I have outside information I can't share with them. Really. Okay, so what do we do to get started in the lab? The interview process is completed by the end of the first full week of classes, and that's really important. So we have to have that organizational meeting, and I have to strike right away. Usually, you know, they call, what do they call it? Syllabus week, right? As the students think of it, that first week. It's still a time when they can add classes. So by the end of that week, my goal is to have chosen them and start the process of adding a course before the add deadline goes away. So that's going to be really the first couple of weeks of January that you have to get all of that in place. By the end of January, the add period will be complete. And then there are several different options for credit, and these will vary by course and also by department. So both of these options require paperwork for you to fill out, and it will be paperwork that the student should bring you, and it's paperwork that they should get from their advisor in whatever program they're majoring in. Okay, so they need to be proactive. Most of these are very motivated, good students, so you don't have to worry about that. They're proactive about going, talking to the advisor, and getting the appropriate paperwork, which they can bring to you. And then there are things for the two of you to fill out. And these are meant to be incredibly helpful in thinking about either the independent study or the directed research options. There's 299 and 299H for independent study, and then 392 for directed research. When possible, I try to get students to go for 392, mostly because this is a better benefit for them in directed research, because it often counts towards the major in fulfilling some requirement for their major. So I will have a 392 number. So for instance, I have a 392 number in my home department, School of Plant Sciences, and I have a 392 in the biology major. I have a 392 in MCB. And I've had 392s in physiology, as well as one of the engineering departments. What is it? Biomedical engineering, thanks, where I've had students. So I've had students from all those, actually in neuro as well, although maybe I had a 299 in neuro and not a 392. Departments will have different policies about what counts towards the student requirements. So ask students so that they know to ask those correct questions. Say, okay, well, tell me, what kind of credit are you getting for this? Are you fulfilling, say, a general elective credit that's, you're getting credits towards graduation, but not necessarily credits to fulfilling your major? Or are you getting credits that will fulfill requirements for your major? And that's going to differ for departments, but they need to know to ask those questions so that they're fully aware of how the credits will be applied. Most advisors are really good at that, but you should know as well that there are these differences, and it won't be the same for every department with whom you interact, as far as my experience.
Mark,
yeah.
At least in our case, is there a situation where independent study would be the better choice?
I'm sure there are times when independent study would be the better choice. I think what I've tried to do is I've tried to make it work for students wherever possible, that they get credits towards the major. And if that isn't an option, then I go for like a general credits kind of option. It's nice because it's molecular biology. A lot of departments across biological science departments across campus will accept those. Okay. The other thing you should know is that 3 credit hours equals 9 hours of research per week. So the minimum that I let students enroll for is 3 credit hours. Because as I mentioned before, it's kind of that critical number. You've got to work about 10 hours a week in order for it to be worth anybody's time. They sign up with a special code that's affiliated with you. You'll have no idea what that is. But one really important thing is, once you finish all the paperwork, which again will require you to talk about you make a little description of the course, what the research will be, and importantly, the deliverables, so how students will be assessed. And that's something you want to give some brain space to anyhow, is how you're going to evaluate them. I'll show you what I do in a second. And then what you want to do after they say, okay, I'm enrolled, everything looks good, is you want to go to View Access Instructor, and you want to check that they appear on your grade roster. don't just assume that all the paperwork went through, because there's a lot of students adding courses, and this is an additional paper form. For some reason, this isn't electronic at all, and so students will, once it leaves your office with your signature, students still have to turn it in, and those advisors still have to process the paperwork. So incidentally, that paperwork just gets filed in the advisor's office. If you want to know what you wrote down, it won't ever appear anywhere else. So make a photocopy of it. So if you want to remember what the deliverables are because they're changing, or you want to remember what the project description was because you need it later for an NSF grant or something, make a photocopy, because that just lives in the advisor's office. It doesn't go anywhere. And then they push the buttons to make sure that the course gets added for the correct credit hours. Okay. And some of these things, I think that depending on how they take it, they can take it for different options too. Like they can take it as satisfactory, like a pass-fail option, or a graded option. I think directed research is always the graded option. And I imagine that 299H is also a graded option, but I don't remember. I don't, too many things to keep track of. I can't keep track of that as well. Okay, so once you actually ensure that they're enrolled by checking U-Access Instructor, what I do is I require each of them. This is something that I mentioned early on during the interview process. I say, I'm going to require you to apply to UBERP. And that's the first thing that we do when they come into the lab. So I match them with their appropriate mentor. So by this time, all of us in the lab have sat down and we've decided who's being paired with whom. And I introduce them, and they start together working on the understanding of the student about the project that they are going to be contributing to. So they'll work with this person that I'm calling the direct mentor. They'll learn about the project. Together, they and the direct mentor will craft a two-paragraph description of their research project. And we will use this in the UBERP application. So once the two of them have talked about that, written the description of the project along with its major goals, I'll read it and revise it, and then it's ready to go into the application. At the same time, students are going to be crafting their personal statement. That gets edited first by the direct mentor, and then it gets edited by me, and then it goes into the UBERP application. So I'd have to say right now I've had about somewhere between an 80 to 90% success rate on students who've applied to UBERP actually getting a UBERP fellowship. And so this kind of workflow has worked really well for us. We identify the really good students, we bring them in, and then that's the first thing they work on. That's job one. And I think it's a really great opportunity to explain to students who are interested in a research career, this is what you do. The first thing you do is you find funding to pay the way. That's the first part of any project. And I let it be known that this is your way to actually contribute. If you get one of these, it's your way to contribute to the lab. And I think that that is a source of pride for most of the UBERP students in my lab. It's like, oh, not only am I here learning and contributing my work and hands to this project, but I'm actually able to contribute on the bottom line as well. So those are due at the end of January, the UBERP application. So it's a pretty quick timeline, as you were alluding to earlier, Jesse, which is right. First week of classes in January, interview process, then add them by the form, and then complete a UBERP application in that time by the end of January. Okay, so what happens after that? They've got their UBERP application in, but we still have a whole semester to get through, right? So they'll work with a direct supervisor. The goal of that is to learn about the project that they're partaking in, right, that they're contributing to, while they're building a set of lab skills that can be used on any project in the lab. So invariably, if students stay for 2 or so years in the lab, they will move projects once or more than once. And I want them to have a baseline set of skills, right? And so that's where their mentors typically focus, is developing those really good bench skills, things, that differs for different labs, but for us, it's things like PCR, gel electrophoresis, as well as interpretation of those data. So we focus on mastering the laboratory bench skills, interpreting those data, and also communicating the results. So at weekly lab meetings, they're all required to bring their notebooks And when the direct mentor talks about results that they have that involve experiments run by the undergraduate, I'll typically ask to see the actual results from the undergraduate and ask for an interpretation. What does that mean to you? To see if there's a good connection between the direct mentor and the undergraduate. So it's not only figuring out, okay, does the undergraduate know what's going on, but to what extent is the direct mentor communicating the reasoning behind experiments. So everyone should be able to say why you did something, not just that you did something. So that's a really good time to assess their communication skills and the communication between the student and the mentor. Yeah.
That applies to the students that join your group, right?
Yeah, this is all what I do. All of it's just what I have done over the last 7 years now.
The students selected by other faculty, you let them work it out.
Oh yeah, I'm not telling anybody else what to do. Okay, so this is it. This is my last one. So this is my strategy for moving on in the lab. Okay, great. I got great qualified students in the door. We've applied for funding. They are building their basic molecular biology skill set while learning about a particular project and being able to communicate about that project. At the end of the semester, when you're working for credit, they have to present in a multi-lab group meeting, and that will be a 15-minute presentation, a scientific talk. They will work on that with the direct mentor, so all the slides they will produce together, the direct mentor and the student. And then those students will present that talk to me, and over the course of, now this is probably where I do 80% of my job in terms of mentoring those students, aside from the occasional interactions throughout the semester. But 80% will be going through this and workshopping explanations. So my goal is that they should be able to, regardless of what the results are of what they did, if they did 3 PCRs over the course of the semester that worked really well that they can interpret, great, okay. But can they sit down and explain to me and to others the logic of molecular biology, how we think about putting together a question and results so that we can move on to the next question, how do we develop the next hypothesis? And so that's what we workshop. I make them go over it multiple times with me, working on specific explanations. This is how scientists would say it. This is how we do it. I want to see as polished a talk as absolutely possible. And again, regardless of what the results are in terms of how groundbreaking it was, is really irrelevant. It's their ability to communicate that. And getting back to that big impact picture for them. So they present in that group meeting, then I consult with the direct mentor on student progress, and then I assign a grade. And students then are assessed in terms of whether they really liked the experience, whether they were productive, and if their direct mentor agrees that they should continue on in the group. So do you like us? Do we like you? Should we keep working together? And anyone who passes that, but which over the last 7 years, I've only had 1 student who left after taking a research credit. And then I get them into a summer position, and these are, again, paid positions, and students can remain in the lab as paid employees as long as the working relationship is good. And the majority of my students leave upon graduation, but not before. And my goal, and one of the most fun things is, if you can keep them that long, by the time they're a senior, then I work directly with my senior students on an independent project of theirs. So that's a direct report to me. So no longer do they go through their mentor anymore. It's okay, we're working on a project together, and we design out the experiments together. We talk about the results together, and we think critically about some avenue that the lab isn't, it's not a side project of somebody else's project. It's some avenue in the lab that we couldn't pursue unless they had these basic skills and they could think critically about data. And so it's really fun to see them build these project skills. This is where I see a huge payoff in terms of that next generation learning to manage a project, right? Because now they are managing their own project to think about how you plan out experiments all the way through to analyzing those data and coming up with the next question. Right? And I mean planning out in all ways. What's the most expensive experiment you could do? What's the least expensive experiment you could do, right? How are we going to think critically about the amount of time it takes to do X, Y, or Z, given the amount of time it takes? All of the project management aspects that if they leave the university with no other really, well, that's not true. They hopefully will leave the university with many, many skills. And what I hope is that they leave the university with these project management skills that are translatable to whatever field they go into. When they walk in the door, they know how to manage themselves and their time and assess the impact of investing in particular types of either experiments or projects so that they can have the most impact possible. Okay, that's all I have.
All right, thank you, Dr. Beilstein. Up next, we have Dr. Rachel Gallery. Take just a moment to get her set up. You need to step out for a moment. Can we put your presentation on the desktop?
Automatically invited to.
Yes.
Oh, yeah. I was thinking actually we should probably do that when we were talking about photosynthesis.
Next time.
Yeah, for sure.
Yes.
Accumulate the fact that they paid through the end is just beyond us. Very exciting.
I am. Nice if they get the message that it's a professional thing, right? Like,
that's the area that's being recorded.
so you should earn money for things that you invest in.
So do you write that into your grants, or is it you write
Absolutely, I
it
read
into
it.
from the beginning, or you write it into grants?
Yeah, so I mean that lasts as long as I have money. But I mean that's the only reason you do projects, because you've got money. You know, NSF is really good about that. So any NSF grant, you can have that REU supplement. That's what I did. So I usually factor them in, and then for additional students, typically they'll get one or two. And with UBER, the amount they'll give for the summer is like $6,500 per student. That money will last all year long. So, and I can get with REU funding, then it's enough to pay at least one.
So the 6,500 goes to the faculty and the student.
So I pay the student like the $12 an hour going rate, but it's done through the grants office. So I mean, it comes into an account, but it's only personnel costs.
Right.
Money can't be used for anything else.
So when they give you that money, is it summer, fall, and spring?
They don't. They basically give you the money and they say. And it actually, I think it expires.
All right, so our next presenter, if you're all ready, is Dr. Rachel Gallery. She's going to go over some basics on how to set up a lab at UA and kind of figuring out that process and managing that as well. So welcome, Dr. Rachel Gallery.
Hey everyone, I know some of you, not all of you. I guess so. First, what I'd like to say is congratulations and welcome to the U of A. You're going to set up a new lab, and that is really exciting and also really daunting. So I'm an associate professor. I've been here at the U of A now for 7 years, and I definitely remember the first day that I walked into my lab space. That was mine, and I looked around and I was like, oh boy, what do I do, right? And so I've made a lot of mistakes, and I'll share some of those, and also some things that hopefully will help you. And so I don't mean to be glib with, and so I've just organized this as a Word document, and so I'll walk us through it and just sort of talk off of it. I don't mean to be glib with this first bullet point, but in terms of setting up a lab, honestly, ideally, you will hire someone who has been a lab manager in a lab that is similar to what you would like to set up, and then you're going to let them take the lead with a lot of input from you. And so before you do that, there's a lot of things that you have to think about yourself, and it really is helpful to sit down and spend some time mapping out what your lab's core themes are going to be, and essentially write your research mission statement. In many ways, this is like writing your tenure document. So the things that, if this was the perfect world, the research ideas that you are going to get through in the next 5 to 6 years, what were the burning, super cool questions that you want to ask and answer, that's going to help you guide what your lab should be set up to do. And so it is worth it to sit out and try to write that mission statement with the full knowledge that is not at all how things are going to end up, right? But you have to start somewhere. The value of doing it this way too is that it also helps you identify the priorities for the grants that you're going to be writing, right? And then which calls and which funding agencies support that type of research. And then you can start to get creative. As you get rejected from one program, you can think about what can be recycled and used in a different way in a different program. But it also is helpful because a lot of the different funding agencies have different restrictions on what you can spend your money on, right? And so whether or not large equipment is something that you can also buy off of for example, there's some NSF programs and others that allow you to put in for major infrastructure grants, or whether you want to invest mostly in personnel. You know, there's a lot of different restrictions depending on what type of research group you're setting up. And I should say up front that I'm coming from this from the perspective of someone who is an ecologist. So I run a wet lab. So the work that we do in my lab is focused on soil ecology, plant soil ecology, microbial ecology. So we do a lot of dirty work. Like I said, we have a wet lab where we're mostly dealing with soils, but we also have a very clean and sterile lab where we're dealing with DNA extractions and fungal isolations and things like that. And then we also have a quantitative space, so a lab space. And I'll get to that in a moment about how to sort of think about setting up your different areas and priorities. So if you've written this research statement and this mission statement and you've put it in there with some priorities for yourself for the next 6 months, what are your grants? 1 year, what are your grants, et cetera, then you're going to know what kind of stuff you need to do in your lab, but you're also going to know, hopefully from some experience too, as a postdoc and as students, the type of work that you personally do not enjoy doing physically, and therefore that you don't want to set up your lab to do. And sometimes that's just as valuable, right? So if you really don't enjoy culturing and keeping vast cultures clean and sterile and hygienic, then don't set up that program that requires your lab to be that host, right? Know who you can collaborate with and do that in a more efficient way. So that was something that I think I learned a little later than I wished to learn. After I had bought 2 really nice thermocyclers for PCR, I realized, you know what, it is actually, for the work that we do, more sort of efficient and actually less expensive for me to just cost that out to another research group, and then we analyze the data. So those are things that you should just really be thoughtful and careful about. You will buy equipment that you won't use. It is a guarantee, right? And you will also always run out of refrigerator and freezer space. That is a guarantee. If you're working in the ecology sciences, plant sciences, that type of thing. So those things are just inevitable. So just plan for that, I guess. So then that does bring me to this, like, so plan for the projects that you know you want to do in-house versus the ones that you want to contract out. And in part, that's your lab identity and your research group identity, because then that's what you become known for. And then when people are thinking about, oh, you know what, there's this one particular extraction that the gallery lab group does really well, right? So then you can then be that resource as a collaborator for other units as well, right? And so there is value in being able to do many different things well, but there's also an enormous amount of value in being expert in one particular type of analysis or one particular type of assay. One of the things that was really helpful for me was to go around when I was giving a seminar in a different campus or when I was visiting a national lab to just ask if I could see someone's office and someone's lab spaces that were similar enough to what I was doing. Because you learn so much, like you learn that you can't put this next to this because it shakes and then you can't use the balance correctly, right? These are all things that if you don't think about it, it will turn up later when you have a whole bunch of errors in your data, honestly. And so taking advantage of where you're visiting national labs and other places can be really helpful. And just ask people candidly, you know, where did you go wrong? What mistakes did you make? Any offer of advice that you have. I think that everyone will be aware of this, but there's maybe a sneaky thing that you're not aware of, but there's always new vendor offers and PI discounts. The U of A has a really good structure for this in place, and then you're gonna be honestly harassed by a lot of the vendors in terms of they really wanna sell you and offer you lots of discounts, buy this in bulk. You're gonna need some things in bulk, pay attention to that. The one thing that you may not be aware of, and I think this is above board, I really honestly do, is that you can set up yourself first as the new PI. And then your lab manager can also be set up as when yours expires, then your lab manager can be set up as the new PI. And then you get that extended discount for a long time, and it really does matter. Having relationships, especially with some of the vendors that you're going to use the most frequently, having their phone number and their email can be super helpful. When they have discounts, when they're trying to offload a whole bunch of products, they'll call you, honestly, and they'll email you and just say, hey, we're trying to get rid. We bought way too many -80 freezers, so we're offering them at half price. Do you need one? That is an example for me. So that happened. I was totally taken by surprise when they were offering a -80 freezer, of which I needed 4, and they were all half price, right? So that was great and lucky. And then you lose in other ways, right? But so win when you can, right? These people, you know, they are trying to sell you something, but it's in your best interest to get the best deal that you can from them. There's been a lot of moves towards, and I'm a fan of this, I do not share space. I have, as I mentioned, I have a large wet lab space, I have a smaller clean room space, and I have a computer workstation space. And those are 3 separate rooms, and I'll talk about that. Those are biosafety and chemical safety reasons. But I don't share lab space, and when I first got here, I was sharing, and I really appreciated that, and I got a lot from it, and I think my research groups also got a lot from it. You're interacting with people who have a different agenda, a different perspective, and so you can have shared lab meetings, and that can work out really well. It does add a whole new layer of interpersonal complexity about how do you share space, who's responsible when a piece of equipment breaks, you know, and those are things that as long as you're very clear and upfront and you're working through, and I'll get to it later, you have like systems for keeping track and keeping people honest, that can work out okay. Or you just have to deal with them on an ad hoc basis as it arises. But honestly, thinking about sharing equipment can be a really, really good way to minimize the overhead on you. So if you buy a large piece of equipment, that's great. You have your startup and that's going to work well for you, and then it's going to run out at some time. And the unfortunate thing is that most funding agencies will not pay for maintenance, right, or for when something breaks. And so you really do run into a challenge where you have to be very creative about how you can use monies in order to just upkeep the very expensive equipment that you have. And unfortunately, the U of A is a little bit cracking down on these contract facility centers. So that's your lab is a place where you can charge for a particular analysis in order to keep the maintenance costs of your equipment down. And so that's that would be great if that could happen. We're currently not really in that position, other than something like the Cancer Center. I think this is a big important piece of advice. Depending on the type of lab that you're setting up, you're going to be under a different biological or chemical safety criteria. And it really behooves you to meet early with the inspection officers, find a contact, make friends, right? It's a pretty challenging process to negotiate, and as long as you're having that open dialogue and discussion about what's exactly going on in your lab, there won't be any confusion, and you won't end up with a biological or chemical safety criteria that's higher than what you need, because those do have consequences for the types of training that are required, the types of lockdown facilities, et cetera, that are required. And so they can really help you determine the level of safety you need. You'll write a series of SOPs, standard operating procedures, for the different types of research categories that you're running. And then they'll use that to identify what restrictions, what permits you may need if you're taking, for example, if you're accepting samples from different parts of the country or even of the world, you're going to need some different types of permits to help with that. And all of that can take a lot of time, and sometimes it can even take some money. If you need some retrofitting in your lab for things that you haven't thought of, if you need areas that need to be locked, for just as 2 examples. And then the oversight, I think that we're a BSL, Biological Safety 2 and a Chemical Safety 2, and we end up having inspections, I think it's every 6 months or so. Does that sound about right? Is that extreme or about right? I think it's about every 6 months or so. And it just helps keep track of all of the new people who come into your lab and all of the training that they have done, and if you have new SOPs, or if you're expiring things and you're no longer need to deal with certain issues. They also help you with all of your waste and how you get rid of different types of waste. And all of these things require a lot of time, which brings me back to this front one here, which is that ideally you will decide to spend some of your startup money on a professional who really can help you set up your lab. You are starting to run a business, right? And so you're the CEO of that business, and you don't want to be doing all of the work. You want to be hiring out the best people and the experts that are going to be taking the lead on certain things. And setting up a lab is a very special skill, and people have that unique skill set and can do it. Probably better than you can, especially if you haven't set one up before, truly. Yeah. You're at the stage where you're at the U of A, so hopefully if you needed any renovations, you have it all in writing. It's too late now, I guess, if you don't, honestly. But if you do need renovations, go immediately and talk with your director and talk about what they can do and what you will be financially responsible and what can be done otherwise. One of the things that I found to be enormously helpful is, so for the folks who are the custodial staff who are cleaning your lab space, sometimes they might be very confused about what's going on in there. They might be concerned about something that they see in there, right? They sometimes are in that space. Maybe there's something that you're not even aware of that they identify as a potential risk or a challenge, right? If you talk with the staff, you walk them through your lab, you show them what's going on, you show them the areas that you've identified as toxic or places that are of concern, ask them what their questions are and what their concerns are. This is enormously helpful. And oftentimes, the custodial staff are dedicated to particular lab spaces, and so they'll be really familiar over time with what your lab needs are, and they can even help you anticipate some of them. Like they can help you get some of the, for example, biological waste bags that you might need, or even just provide you with extra cleanup supplies if that becomes necessary. And so, as I had already sort of mentioned, with your startup money, and we'll get to that when we talk about the budget, try to squirrel some of that away if you can, for the upkeep, for the equipment that you're going to identify that you need to buy, and also for new computer purchases and software. Again, there are a lot of funding agencies now that do not want to pay for computers or for software unless you can prove that those are going to be dedicated to that project only, which is a real challenge to do, right? And so that's what some of the monies for your startup are useful and valuable for. I don't know if there was anything else that you maybe wanted to include, Mark, or anyone else about how to set up a lab. I guess I've identified mistakes that you will make, buying equipment that you don't need, organizing a space and then realizing you need to change the whole thing. That will certainly happen. I don't know what else there is. Well, one of the things for me that was helpful, our lab is in the basement. And so when one of our DI water faucets broke and was shooting an enormous plume of water at maximum volume into the ceiling, we were all looking for the shutoff valves along sort of under the sinks and along the floor and all around. sort of not paying attention to the fact that we were in the basement and all of the shutoff valves were actually in the ceiling. So that took about 20 minutes for us to identify and find, and that was a big mess, right? So that's one of those things where it's good to know where your shutoff valves are. You're welcome. So think about that. And then also when you use the eye wash stations, which, you know, hopefully you won't have to, but it's likely that you will, those are also sometimes designed to flood a space, right? And so then you'll have to deal with the cleanup of the flooding after that. So those are just some practical things about setting up a lab. And then just a few thoughts on the budget and on hiring, which is, we'll spend less time on this and a little bit more on the budget part, because the budget part is honestly very daunting. And you might think, oh, I can just rely on those financial reports that I get monthly from the U of A system and everything will be great. And I really need to emphasize strongly that you cannot. That there are frequently errors and they are never in your favor. And so you do need to really pay attention and be on top of your own budgets. I keep a separate sort of spreadsheet system for each one of my budgets, and I know everything about it in terms of when they expire and the 6 months before that, so that you know how and when to be spending down when you need to. In terms of your startup money, they often come from different pockets around campus, and they have different expiration dates. So you also need to think about that and sit down with your business office and say, please identify for me what expires first, whether there are any other restrictions about how you can spend the money. Sometimes you can't spend it on personnel. Sometimes you can't spend it on equipment. And so just make sure you have a great plan in place for that. Really do keep track of all your grants independently of the UA system. It's great to check in and have a formal business meeting with your business office. I mean, if you can do it quarterly, wonderful, or even once per semester, minimum. Do not leave it to annually, or worse, don't ever not do it, right? Check in. It is confusing. Even when you're sitting down with your business office, it's confusing. And ask a lot of questions, and really do just never leave the room without an understanding of what's happening with your budget. It's your budget. It's your business, right? It's your lab. And so don't assume that anyone else is looking after this for you. This really is, you are the CFO. You are the Chief Financial Officer for your lab. And we are lucky. I'm in the School of Natural Resources. We have an excellent business office. And even with them, I sit down and I have questions and I'm confused and I really can't work out. So how much money do I really still have left in this grant? It says negative 20,000, but you're telling me I have 30,000? Which one's true, right? And the answer is somewhere in the middle, right? So do check in with your business office. The software that we like to use, just in terms of interacting with the lab, we like to use Quartzy for ordering things and for keeping track of resources. It's a great system for doing that. The other nice thing is that it interacts with UA byways, and you can make purchases through it now for some things, not for all things. But it's also a way that your lab manager can help you keep track of what you're running out of in terms of, it has a full chemical inventory, but also a disposables inventory. So it really is helpful. Nice thing about it too is that it has all of the ordering codes in it for you, so that you're not, so it's like, you know, I know that we wanted this particular type of whatever. You can order the exact same thing. So we like Quartzy, but there's lots of them. And so I'm just going to refer to that one because that's the one that we've used. Okay, so just to wrap up a few final things, and I'm not going to spend too much time on the hiring, but just a few things that have come up for me. The first one, I think, is don't panic about the hiring, truly. And don't hire people to fill your space because you think, I need people. Oh my gosh, it can't just be me in my lab. You know what? It can be you in your lab for as long as it needs to be until you get the people who fit with your lab, right? Who fit with they have the shared interests. Mark has already sort of identified all sorts of wonderful ways of trying to identify students to recruit into your group. I think demonstrating independence and empathy are really important parts of who your first hires are going to be, because they set the whole tone for your research group. And you know, mistakes can happen. Be thoughtful about how to deal with those mistakes. Have a 6 month
Yeah.
Probationary period.
Yeah, probationary period where, you know, you can say, if it doesn't work out after the 6 months, then we're going to part ways and no hard feelings. And work that into a contract that's really honest and open and upfront. This first one at the top, I've seen a number of people do this, and I think it's wonderful. Include whatever your lab mission statement is on your lab website in the place where you're saying, we're recruiting people, so that people really know what is the tone of your group and why would they want to come work with you? Not just a research statement, but a diversity and inclusion statement. That can be a really valuable way of attracting the people that you want to attract initially before you even do some further screening about whether their research interests are compatible or not. Obviously, ask your colleagues and former advisors to send promising students your way. As Mark was mentioning, recruit students from your class using his wonderful criteria. You can volunteer to give guest lectures in classes where you know that there are going to be students who are going to be interested in your work, right? So go out there and volunteer and make your research pitch in that class, right, to try to capture those students as well. And more people are using science Twitter to find ways to go to grad school or to find undergraduate research programs, take advantage of those, and meet with people at conference meetups. So it's hard often to get students to come out. Especially at the grad school level, it's hard to find money to get students to come out and visit, but it really is important to have that face-to-face interaction to really determine whether or not you're gonna be compatible, because that relationship is foundationally important for a graduate research program, I think, and postdoctoral as well, and undergraduate as well, but especially at a grad level, when you have a student who's committing to work with you for 3 to 5 to more than that number of years. And so finally, these are 2 books that I have found to be helpful, that you may find to be helpful as well. Radical Candor, for me, was really useful because I realized that I wasn't doing that, and I was really doing my students a disservice by being too kind. So Radical Candor is the ability to challenge directly, but also show that you care personally about a student's success or anyone's success. So it is hard to challenge someone directly in a thoughtful way, right? And to not make it seem like you're trying to undermine or undercut them. And we all deal with negative feedback in different ways. And so this book, Radical Candor, was helpful in helping me identify where I was essentially failing as a mentor to the students in my group. What I was doing was erring on the side of demonstrating how much I cared personally about their success, and in so doing, not wanting to hurt their feelings by giving them much needed advice that was honestly and truly going to help them. You don't get better if people aren't pointing out where your weaknesses are, right? But at the same time saying, here's where I think you can work on yourself and do better, or work on this approach to research and do better, and here's how I'm going to help you do that. Right, so not just leaving it as so good luck with that, right? Let me know how that goes. And then the 5-Minute Manager is a very small and short book. And again, it's a way of having quick check-ins with your mentees where everyone leaves the meeting. The takeaways that I have is that everyone leaves the meeting with a sense of, here's what we've accomplished, here's where we're going next, here's some parts that were uncomfortable, but we're leaving on hopefully a positive note with some positive action plans, right? We've found some solutions. We haven't just identified the problems and feel sort of miserable and bad about it and beat down. So, and it's a short book and quick and easy to get through. And then finally, there are lots of resources, but one of the resources for lab setup that I have found to be helpful is that from the Howard Hughes Medical Institute. And so this is a specifically lab setup resource. So walking you through just a lot of the nuts and bolts of how to set up your space, essentially, in a way that's going to maximize your efficiency and minimize issues, especially issues related to accidents or health and safety type issues. So that is what I have in terms of some things to get you started and things to get you thinking about in terms of setting up your lab. And I really do, like I said, recommend those 2 books because you really now are going to start thinking of yourself and your students are going to start thinking of you as someone who has answers, right? And you don't have all the answers, right? And so the ways in which you can set up that dialogue with your students early, with your lab manager early, and your postdocs early, to show that you're going to be searching and finding these answers together, I think the earlier you can set that tone, the better off things are going to be for your research group, as people are going to take ownership of their value and of their position within your research group. So thanks for your time, thanks folks, and good luck to everyone who's getting their lab set up.
Thanks, Rachel.
Rachel and Mark, are you willing to share materials that you can?
Yes, I mean, absolutely,
Yeah.
especially the books and stuff. Yeah.
Thank you, Dr. Beilstein and Dr. Gallery, for coming and taking some time to present. I know we have a little bit of a small turnout here, but we'll make these resources available online so they can help out other faculty members who aren't able to join us today. So thank you so much, and enjoy the rest of your week.
Great, thanks.
Sure.
Yeah, yeah, exactly, yeah.
That's great.
I could use that as like 7 years ago.
Oh my goodness.
But don't you like the paid forward aspect of the work? You're like,